Alright, next up we look at structure question 3. Right, so the limiting factor in the zone marked X obviously is light intensity, because for X, right, when light intensity increases, the rate of photosynthesis also increases, right? So whatever increases causes the rate of photosynthesis to also increase. That would be the limiting factor. So in this case, it's light intensity. As for Y, right, you can see that light intensity is no longer limiting, right, because light intensity increases, but the rate of photosynthesis does not increase anymore, right? So we can say that the light saturation point has been reached, right? So light intensity is no longer the limiting factor. So another factor is limiting now, and from the graph we can conclude that it would be temperature that would be the limiting factor, right? So instead of leaving it vaguely, like so vague that another factor is limiting, full stop, right? You can conclude which one is it from the graph. Okay, so temperature is the one, because you can compare graph A and B and see that as temperature increase in graph A, right, the rate of photosynthesis also increases, right? So between A and B, we can conclude temperature is the limiting factor when the light intensity is high. Okay. Okay, then move on to part 3, right? So for part 3, it tells you about the saturation point, which is 100 W meter square. Okay, because it says here the plateau, right, in with the rate of photosynthesis is reached. So this plateau would mean that this is the light saturation point, right? So the light saturation point is about 100 W per meter square, and they tell you that the usual amount of sunlight would be 1000, which is much higher than 100. Okay, so obviously this tells us that the light intensity from the usual amount of sunlight is much higher than the light saturation point. Therefore, light intensity is usually not the limiting factor. Okay, usually, right? However, there will be instances when light intensity can be the limiting factor, because the question, the information tells us this 1000 W per meter square is only for a clear summer day, right, at midday. Okay, so when it is not clear, when it's not summer, and when it's not midday, right, then the light intensity can be quite low, right? You can imagine, such that light intensity may become limiting. Okay, right, so there's no mention of the other limiting factors here, because if you read the question, it says, what does it suggest, right, about light as a factor. Right? So the focus of the question is just on light. That's why there's no like mention of the other limiting factors and suggesting that they may be limiting. Right? So we're just focused on light itself, whether it is limiting or not, right? When is it limiting and when is it not? Okay? Okay, then move on to part B. Right, so it mentions here, right, for the two different temperatures, light intensity is the limiting factor when the light intensity is low, but light intensity is no longer the limiting factor when it is high. Right? Basically, that's what the question suggests, and that's part of the answer as well. Right, so the rate of photosynthesis is the same despite the changes in the temperature when light intensity is low, because light intensity is the limiting factor. Then we give some explanation how light intensity limits the rate of photosynthesis over the temperature. Okay, so the rate of Kelvin cycle specifically, right, is slowed down by the lack of ATP and reduced NADP when light intensity is low. Right, so even though the let's say in 15 degrees Celsius the temperature is lower, right, the enzymes are working slower when temperature is lower. However, the lack of ATP and reduced NADP is like even more significant than the enzymes being slower. Okay, so that's why the limiting factor is light intensity and not temperature. Right, so even when the enzymes are, whether the enzymes are slower or faster, right, Kelvin cycle is just as slow because of the lack of the ATP and reduced NADP. Okay, but obviously the reverse is true for high intensity, high light intensity, right? So for high light intensity, right, we got to first mention, as a contrast back to our previous point, right, that there is sufficient supply of ATP and reduced NADP. So that's why the light intensity is no longer limiting, because now we have sufficient supply of the resources for Kelvin cycle. And now Kelvin cycle is limited by how fast the enzymes are working. So that's why it's temperature that is serving as the limiting factor. Right, so this question is not just asking you to regurgitate the effect of light intensity versus effect of temperature. There is a bit of the requirement for you to kind of compare between the significance of the limiting factors, whether it's in low light intensity or high light intensity, right? Okay, let me try to be clearer here. Because for limiting factors, right, a plant is always subjected to the different limiting factors, like all the time. But there's always a particular temperature, there's always particular carbon dioxide concentration, there's always a particular light intensity, right, that the plant is subjected to. Right, so all the limiting factors are in play all the time. It's just that at any one time, one is the most limiting. So there's a bit of comparison here. So just like I mentioned just now, for low light intensity, the temperature is different, right? So the enzymes are working in different speed. So for the one in 15 degrees Celsius, the enzymes are slower, but that is not the main reason for why photosynthesis is slow. The main reason is the lack of ATP and reduced NADP, right? So it's not just about explaining one limiting factor at one time, right? You don't just regurgitate the effect of one limiting factor. There's a little bit of, in your writing, right, the idea that one of them is the most limiting, the most significant, but that idea needs to be present in your answer. That's why in the answer for high light intensity, right, it is important for me and for you, of course, right, to actually mention that there is sufficient supply of ATP and reduced NADP, so that it shows as a comparison that, okay, the light intensity is not the limiting factor here. It's not so significant compared to the speed of the enzymes, versus in low light intensity, right, the speed of the enzymes is not so significant. It is the lack of ATP and reduced NADP that's more significant. So a bit of the idea of comparison of significance in the limiting factor is required for a good answer. Okay, so this is the difficult part of the concept of limiting factor. Usually we only think of one, right? Intuitively we would look at only one and then just regurgitate the effect of one. But to fully answer the concept of limiting factor, right, we need to remember that, yeah, all the limiting factors are in play all the time, but one is the most significant. So we actually have to mention all the limiting factors, but in our answer to explain that one is the most limiting. Yeah, okay, instead of just regurgitating on one and not mentioning the rest. Yeah, okay. Okay, let me just continue, yeah. For part C, okay, so for part C, it talks about the carbon dioxide concentration and the average atmospheric carbon dioxide concentration is 0.035%. So what can you conclude, right, from this? Okay, so the graph, right, shows you that carbon dioxide concentration is the limiting factor between 0.01% and 0.4% when you compare graph A to C. Okay, so now between graph A to C, right, when carbon dioxide concentration increases, the rate of photosynthesis increases as well. So this shows you that we haven't reached saturation, right? So within this range, where carbon dioxide concentration is the limiting factor. Okay, so 0.035 is within this range, right? So therefore, we can conclude that 0.035% is usually poses as a limiting factor for photosynthesis, assuming that there is sufficient light and the temperature is optimal, around 25 degrees Celsius. So again, back to the same concept of limiting factor, right? Remember that the plant is subjected to all the limiting factors at all time. But at any one time, one is the most significant. Okay, so when you want to conclude that carbon dioxide is the limiting factor, meaning carbon dioxide is the most significant factor, then you got to mention that the other factors are not that significant. That's why it's important to have that third point to say that there is the assumption that light intensity and temperature are not limiting factors for us to conclude carbon dioxide is the limiting factor. All right. So the same concept of looking at all the limiting factors, right, and a bit of comparison between which one is the most significant. Yeah, is required here instead of just talking about carbon dioxide itself. Okay. Then move on to part D. Right. The external factors, quite simple. Right. You can talk about water. You can talk about oxygen or magnesium. Okay. So I think water and oxygen you are familiar because as I went through with the other classes, they are all okay. So I don't think I need to explain too much for water and oxygen. For magnesium, you may not know this. For magnesium is found in chlorophyll. Right? So it's needed for chlorophyll synthesis. Okay. The question is external factor. Right? So we are looking for things that are absorbed from outside the plant. So if you suggest chlorophyll concentration, right, then that wouldn't be an external factor, right? There's a bit more internal. Okay, so magnesium would be a better answer for that. Okay, so just a bit of chlorophyll structure for you to see that there's the magnesium right in the center of the chlorophyll plane. Right, we call it the porphyrin circle. Or porphyrin ring, right? So magnesium is in the center of the porphyrin ring there. Okay, yeah, and it's just a comparison between our chlorophyll porphyrin ring as well as the heme group in hemoglobin. Right, so actually they are quite similar. Yeah, in fact, very pretty much identical. The only difference is one has magnesium, the other one has iron. Yeah, so I thought this interesting. Okay, and the last part, right, products of the light-dependent stage. So obviously it's ATP, reduced NADP, and oxygen, right, as the byproduct. So don't forget the oxygen as well, yeah? Okay, well, just one last extra question. This is from a past year CT1 essay. Okay, so the question talks about explaining, right, what is meant by the term limiting factor, but using temperature as the example. All right. So I just want to use this question to again, right, repeat what I tried to explain in terms of the concept of limiting factor. Right, you cannot just look at one factor. Right, you look at all the factors. So in this case, right, in this question, what happened was when the students did this question last time, right? Most of them failed this question. Why? Because most of them only talked about temperature. Okay, so imagine okay, you draw a graph to try to help you, right, in to answer this question. What kind of graph will you draw, right? So what happens is most students do this graph, or they explain this graph. The explanation kind of is about this graph. So they just regurgitated the effect of temperature. But that does not explain what is meant by limiting factor. Remember, limiting factor, we must look at all the factors and then compare them in terms of their significance, and to prove or to explain that one is the most significant. Right? So the answer to this question would actually require this graph instead, the one on the right, to compare all the different limiting factors to explain when temperature is the most significant. Then you regurgitate the effect of temperature. Okay? So this is what I meant, right? When I mentioned the concept of the limiting factor, right? You shouldn't just look at one. You need to look at all of them. Have a bit of comparison between them to explain which one is the most significant. So that would be the limiting factor. Okay? I just wanted to use this essay question as an example to further highlight what I was talking about. Okay? I hope it's clear to you. All right.